In this video, we are going to talk about some statistics that I want you guys to know for this class. Now, this is not supposed to be a statistics class, so we're not going to spend a ton of time on it, but just some terms that you should be familiar with and that you're going to see as you read results sections. Our learning objectives are to compare and contrast descriptive versus inferential statistics, to determine when to use a statistical method and pick an appropriate one, to define some key statistical terminology, to describe common comparative versus relational statistics, and to be able to write about statistics in results sections of lab reports. There are two major types of statistics. The first is descriptive, which describes the distribution and the tendencies of the data. So what does the data in front of you look like? What is the mean, otherwise known as the average? What is the median, mode, range? Median and mode, remember, are ways of saying the middle points. The range tells you about the scope, and then the variation will tell you about how distributed the data is. So variance, standard error versus standard deviation. Inferential statistics, on the other hand, are used to infer properties of a larger population based on a sample. When you run an experiment, you're not testing every single person or every single mouse or every single worm that exists. You're doing it on a smaller scale, and you're hoping that your sample is representative of what would be happening in a larger group. And inferential statistics can tell you whether that is true or not, and give you some idea about probabilities related to what you see in a group, comparing it to a larger population. And those are things like t-tests, chi-squared tests, ANOVAs, and correlations. Why even bother with statistics? Well, we need to use statistics to analyze data because there is natural variation in biological populations that makes it difficult to determine actual means, actual differences between groups, especially when we talk about humans. Humans are incredibly diverse, and trying to figure out what is considered normal or average is a challenge. We are almost always examining a sample of a greater population. As I said, it's almost impossible to do this on every single person or every single mouse. And so instead, we're testing a small sample. And we need to think about that sample when we want to compare our results to a greater population. Statistical analyses essentially help us determine if our data are reliable enough to reject our null hypothesis and accept the alternative hypothesis. So to reject the fact that the independent variable and dependent variable are not related and accept that there is a relationship between those variables. Stats help us determine the likelihood of actual relationships in our data sets as opposed to these things happening by chance. They can't tell you if you found the true mean. So it can't tell you if that's actually what the average is in all humans or all mice or all trees, but it can give you some power in convincing yourself that the data is more likely to be true. So we have two data sets that we collect. Let's say average height of a plant in centimeters. We have data set 1 and data set 2. You can see that it's not always the same, right? We have some variation. And you want to ask yourself, are these two data sets similar? Is the means similar? And if they're different, how different are they? So they both add up to 450 if you were to sum them. They both have the same mean, right? The same average. They also have the same median. But the range is very different. In data set 2, they're more tightly clustered, versus data set 1, they're more widely distributed. Does that matter? All right, the standard deviation, again, showing you the difference in range here. So statistics can help you determine if that difference is significant. These two samples are significantly different from each other. Before we talk about how we figure that out, we got to throw out some key terms here. Common statistical terms that you should know. The confidence interval is a value you set before you start your project. How confident do you want to be in your statistics? Most of the time, we set our alpha at 0.05 in biology, which represents a 95% confidence interval. However, if you're working for a pharmaceutical company, you might want to set your alpha even lower. You might want to be 99% confident, and so you would set your alpha at 0.01. Just depends on the experiment you're running and your threshold. The probability value or the p-value is a metric that indicates the probability of getting your results if the null hypothesis was true. So that's getting the results that you see just by random chance, not that the independent variable is affecting the dependent variable, but just randomly by chance, you got that data. In biology, again, usually we set our p-value at less than 0.05, which is our alpha value. See how those numbers are related to each other? Usually is enough to reject our null hypothesis. But again, it depends on the situation that you're working in. Statistical power is the probability of accurately rejecting the null hypothesis when it should be rejected. A low statistical power can prevent you from detecting a difference when there is one. Power goes up with increased sample size and decreased variation in your data set. So if you have, if you're studying genetically identical mice, for example, clones, and you're looking at the effect of a drug in those mice, you probably only need a group of about 20 to see a difference because the variation is going to be pretty small because they're all genetically identical. You can get a small sample set and see, does the drug have an effect? Is the independent variable affecting our dependent variable? And you can usually tell in that small group because, again, there's not as much variation. And so when there's low variation in the data set, you don't need a large sample to have statistical power, to have enough evidence to reject your null hypothesis. However, when you start moving into situations where there's a lot of variation in the data set, like with people, humans, you're going to need a large group. This is why they run clinical trials with tens of thousands of people in them for phase 3. You need a large group to be able to determine if you're accurately rejecting your null hypothesis because people are so different from each other. There's so much variation in that data set. Okay, so with increased variation, you also need to increase your sample size to have statistical power. Statistical significance. So we always say a relationship or difference is significant when the statistical analysis indicate that the probability of rejecting the null is 95% or higher. So if you hit that p-value of less than 0.05, you can say these are significant statistical analysis. Okay. You also need to know the specific values for the test that you're looking at. Some common stats that I want you to know. So for comparative statistics, we're looking at 2 groups or 3 groups. The most common you're going to see is a student's t-test. And this compares 2 different groups. It says, is group A different from group B? And you can either have one-tailed data or two-tailed data, depending on the distribution. If they're a normal curve, then the curve has 2 tails and they're both evenly spread out, then it's a two-tailed analysis. Whereas if they are a one-tail, when you graph it, it's a one-tail analysis. So for all of this, the best thing to do first is actually to make a histogram to graph the data and see what does it look like, okay? If you've got more than 2 groups, that's when you're going to do an analysis of variance, otherwise known as an ANOVA, which looks at the differences between many groups and tries to figure out what is, which component is causing the variance in that data. Okay. You can also do something called a repeated measure t-test, which you compare for differences while controlling for individual variation by comparing to an individual to itself before and after treatment. This is sometimes called a paired t-test. So you take one person and you measure something before, and then you give a treatment and you measure them after, and you want those 2 values to be compared to itself. So it's a paired t-test. Okay. Your ANOVA, you can think of it like a super t-test. It only tells you if there's a difference between the groups, however. You then have to follow it up with another test to figure out which group is actually the one that's causing the variance. And then you could do repeated measure ANOVAs as well. Okay, relational statistics. This is our correlational analysis. So this examines whether 2 data sets and determines if there is a relationship between them. Usually we do a linear correlation analysis, and you can either be positive, so as one goes up, they both go up, or it can be negative, as one goes up, the other goes down. Keep in mind for correlational analysis, this will only tell you if the things are correlated to each other, not if they're causative, right? So for example, if we're looking at COVID hotspots back in April, they lined up really well with cell phone towers. Well, that's because COVID hotspots line up really well with population, not because cell phone towers are causing COVID. You can also do a regression analysis, which is an R-squared, and this examines the influence of an independent variable on a dependent variable. And often, again, linear, but there are other relationships, and it can help you figure out what factors are affecting your dependent variable. Okay, I said we always got a graph. What we're going to focus on for the homework is the student's t-test, which is again comparing between two groups. This is the formula to figure out your t-statistic. You take the mean of one minus the mean of the other, and then you have to account for the standard deviation with the square root, as well as the sample size in both. Now, when you graph your data, the probability density is the function of the student's t-distribution. And then these are the areas of our tails, okay? Now, this is usually you want the probability, if we're graphing a probability function, this middle area to be 0.95 or 95%, and then our two tails are going to be 0.05. Now you can move this bar and again look for 0.01, 0.001, but usually we keep that at 0.05. When we write about statistics, we want to report that in the results section. And we report the p-values at the end of our first sentence making a claim. So if you say high sodium diets lead to significantly greater blood pressure in adult blue whales, p equals 0.024. Trends suggesting contrasting competitor body color cause increased aggression in beta splendens. However, no statistical difference was found. P equals 0.745. You want to report the actual p-value, not just the relative. So you don't want to say p is less than 0.5 or p is greater than 0.10, unless it's really, really small, unless it's p is less than 0.001. Then you don't have to keep going with all the zeros to get to your actual number. If you're reporting a comparative test, you want to report only the p-values. Other test statistics are unnecessary for this course, so I don't need the full matrix or anything. What we're going to do is we're going to look at M&Ms, and we're going to see the effects of sample size. We're going to compare means to a known population, and we're going to compare the means of two samples. Now for this, I have set up the Excel for you, and you want to watch the next video to see what you're doing and what goes where in the Excel.